everyone. So I think that New Year's and Christmas holidays wouldn't be those if I do not post a new presentation and a new lecture in a new year 2021. So anyway, today I have chosen one topic which most of the students find rather tricky to solve, and that is the story, as you can see, on the metabolism of arachidonic acid. And those are actually eicosanoid molecules. We're going to discuss about their types and metabolism. I'm just going to remind you that those are prostaglandins, thromboxanes, and leukotrienes, and some other novel eicosanoids. We're going to discuss and follow the pattern as you're used to already. So this pattern is going to give you first an overview on the story on eicosanoids, and I'm going to tell you some of the general information about this group of molecules, and then we're going to move on to talk about those specified eicosanoids, and we're going to discuss about their structure, about nomenclature, about biosynthesis, biological function, metabolism, and possible application in medical practice at the very end of today's presentation. So let's start with an overview on eicosanoids. So we can freely say that eicosanoids are molecules which are among the most potent regulators of the cellular function, and literally every single cell in our organism actually is capable of synthesizing considerable amounts of eicosanoids. I have already mentioned that those three major groups of eicosanoids are prostaglandins or PGs, thromboxanes or TXs, and leukotrienes, LTs. Some of the authors claim that those molecules may also act as local hormones, thus regulating numerous processes in the cell. Eicosanoids participate really in a significant number of different processes, biochem processes in our organism. So I'm going to give you an overview, and I'm going to give you a list of those in the overview chapter. So particularly, this is the first thing that comes to our mind, is that eicosanoids are involved in inflammatory response, so which appears after infection or injury of the tissue. So I have just summarized a definition of inflammatory response for those students who are beginners in biochemistry. So this is the sum of body's actually efforts to destroy invading organisms and to repair existing damages. Also, eicosanoids are involved in the control of bleeding. So they are involved, especially thromboxanes, in the formation of blood clots. The overproduction or increased production of eicosanoids may lead to the development of numerous symptoms such as pain, swelling, and fever, which are very important signals for our body to start fighting and to start activating regulatory mechanisms, which responsibility is going to be to diminish the effects of invading organisms and to repair damages. Also, exaggerated or inappropriate expression of the normal inflammatory response may occur in individuals who have allergic or hypersensitivity reaction. But this is not the end of the list. So I have just started, and I would like to give you a little more information. In addition to these functions, we can say that eicosanoids are involved in the regulation of smooth muscles contraction, particularly the contractions of intestine and uterus. Also, they are involved in the increase of water and sodium excretion from our body by kidneys and urine. So they are kind of involved in the regulation of blood pressure. Also, Acorsinoids frequently serve as modulators, which means that some Acorsinoids exhibit stimulating actions, thus another Acorsinoids may exhibit inhibitory action of the same process. And I'm going to give you an example to illustrate this. For example, some Acorsinoids will be shown to have an effect on vasoconstriction of blood vessels, some of them of vasodilatation. Some Acorsinoids will be responsible for bronchoconstriction, some of those from bronchodilation. So we usually say that they kind of act like antagonists. The common characteristic of all Acorsinoids is that they are produced, all of them are produced from polyunsaturated fatty acids containing 20 carbon atoms. Those fatty acids are usually found in phospholipids in cell membranes, so esterified with glycerol in the molecules of membrane phospholipids. And the most important of all fatty acids which are involved in the biosynthesis of Acorsinoids is already very well known to you, Arachidonic acid, which is either derived from the diet or it can be synthesized by certain number of chemical modifications which occur in our body, and the starting point is the molecule of linoleic acid, which is also an essential fatty acid. So all of those Acorsinoids, or at least I can say most of them, are produced from arachidonic acid. I have made a moment of break because some of the Acorsinoids may be produced from some other fatty acids, not the arachidonic acid, but the production of those and the concentration of those occurs to a much lesser extent, and they are kind of a little bit less represented in our body. I don't want to say less important, so I'm gonna mention that in a couple of minutes again, but we're not gonna discuss about those. So anyway, let's start. Arachidonic acid, we said it's a precursor for the biosynthesis of Acorsinoids, and it's enzymatically transformed or metabolized by three major metabolic pathways. The first one is, as you can see, the Cyclooxygenase Pathway, which is actually responsible for the production of prostaglandins and thromboxanes, and Lipoxygenase Pathway, which yields up the production of leucotrienes, and Cytochrome P450 pathway, which generates Acorsinoids with less well-defined physiological functions. As we proceed with our lecture today, I'm going to tell you a couple of words about Isoprostanes, which are molecules which belong to a relatively new class of Acorsinoids, again derived from arachidonic acid, but in the series of reactions which are not catalyzed normally by enzymes, but those reactions are catalyzed by the action of free radicals. So it usually appears when a cell is exposed to oxidative stress. They are kind of similar to prostaglandins, and actually they may play a certain role in inflammatory response, which is caused and initiated by free radical reactions. Some authors also suggest that levels of in blood and urine of isoprostanes can be used to assess or estimate the level of oxidative stress in patients. And one more molecule belonging to the group of endocannabinoids is going to be one of our topics almost at the very end of today's lecture, and the molecule which is first isolated and described in our nervous tissue, especially in the brain, which represents nothing else but the arachidonic acid, which is actually coupled with ethanolamine. And this molecule actually exhibits pretty similar properties to cannabinoids. So it's capable to bind to cannabinoid receptors CB1 and CB2, thus exhibiting biological functions which are similar or analogous to delta-9 tetrahydrocannabinol or THC. Most of Acorsinoids actually have a very short half-lives, so they are active for a couple of seconds, a couple of minutes, and then they are inactivated or metabolized. So we are gonna discuss about those pathways of inactivation as well during our lecture. But of course, we're gonna start with a precursor. We're gonna start with arachidonic acid, which is a source, the major source of Acorsinoids in our body. So we can say that arachidonic acid is definitely the most abundant and the most common precursor of Acorsinoids. What does that mean, Acorsinoids? Let me remind you of your chemistry course. Akorsa is the Greek word expressing the number 20. So Acorsinoids are molecules derived from fatty acids containing 20 carbon atoms. And from previous chapters, from chapter on lipids, I think that you are pretty familiar with arachidonic acid and its properties. So what you know so far is that arachidonic acid is not only essential fatty acids or partially essential fatty acid, but it contains 20 carbon atoms and 4 double bonds. Those double bonds are positioned at C5, C8, C11, and C14. So this is something that is very familiar to you. Usually, also an information which is known to you is that arachidonic acid is usually esterified in the molecule of phospholipids and predominantly at the position number 2 of glycerol molecule. So it is a normal and common constituent of membrane lipid bilayer. Regarding the fact that arachidonic acid is an essential acid, it cannot be synthesized de novo in our body, but it can be obtained from our diet in the form of arachidonic acid or in the form of some other polyunsaturated fatty acids like linoleate, and then it can be subjected to certain chemical modification to produce arachidonic acid. Plant oils are especially rich in linoleate and polyunsaturated fatty acid. So actually the dietary habits of a person can significantly affect the composition and the content of fatty acids in phospholipids and in cell membranes. So people who are taking predominantly plant oils and using plant oils and polyunsaturated fatty acids in their diet will have a higher content of those in their phospholipids in membranes. Oppositely, people in which diet saturated fats predominate, they're going to have less quantity and less amount of those in their phospholipids. But let's take a look at this figure, which I really find nice and representative to show us the overview of the metabolic pathways of arachidonic acid. So let's start from the top of the figure. So we have diet, which should be rich in essential fatty acids, and some of those, like linoleate, can be converted to arachidonic acid, or arachidonic acid itself can be found in our diet. Then we said that arachidonic acid is predominantly esterified to glycerol moiety of phospholipids molecule at the position number 2, which is represented here as one of the phospholipid molecules in membrane phospholipids. For example, phosphatidylcholine is the molecule which is represented here. I'm sorry. And then again, an information which is probably pretty familiar to you from the previous chapters is that certain enzymes are involved in degradation of phospholipids. Thus, we mentioned that there are phospholipase A1, A2, phospholipase C, phospholipase D, and those phospholipases are also going to have an active role in metabolism of arachidonic acid as well. So one of the common examples is that phospholipase A2 actually is active in removing the fatty acid from the position number 2 of the phospholipid molecule, which means that by the action of this enzyme, we're going to release the molecule of arachidonic acid. So here we have the molecule of arachidonic acid written in red colored pencil. Also, we can notice from this figure that glucocorticoid molecules and corticosteroids in general are potent inhibitors of the activity of this enzyme. So thus inhibiting the phospholipase A2 action, we can prevent the release of arachidonic acid and its conversion to different Acorsinoids, thus inhibiting the inflammation in different tissues. So once when arachidonic acid is released from phospholipids of the membrane, it can be chemically converted to different molecules. Cyclooxygenase pathway yields the production of prostaglandins of different types like PGE2, PGF2, PGA2, PGI2 or prostacyclins. And prostaglandins can further transform to give thromboxanes, from which thromboxane A2 is the most common and the most famous. Following the lipoxygenase pathway, arachidonic acid can be converted to leukotrienes, and the example was given when leukotriene A2 is produced. A4 is produced as the first leukotriene, which can be further converted to some other leukotrienes. And finally, arachidonic acid, which can follow the Cytochrome P450 pathway to form different epoxides with biological functions to still be elucidated. So, but let's start from the beginning and let's see exactly in much more details how arachidonic acid can be released from a cell membrane phospholipids. So actually, we have already mentioned from this slide that it can be removed from phospholipids by the action of phospholipase A2, but it can be released also by the action of another phospholipase, which is phospholipase C. The activation of those two enzymes can occur as an answer to a binding of different stimuli to specific receptors on the cell membrane. And again, I'm going to give you one figure which nicely and illustratively represents the process. So what do we have here? We have here different stimuli which can be attached to specific receptors at the surface of a cell membrane. Those can be histamines in allergic reactions. Those can be cytokines in certain infections and infective processes and so on. So anyway, they bind to a specific receptors to that specific stimuli. And the binding to this receptor actually causes the activation of those two enzymes. Usually the activation occurs by the activation of CAMP and activation of protein kinase A, like we mentioned for numerous other processes and regulation of metabolic pathways. But in the very end, these processes end up in the activation of either phospholipase A2 in the pathway marked as 1 and or activation of phospholipase C marked with number 2. So let's follow those two pathways. So from phosphatidylcholine, by the action of phospholipase A2, arachidonic acid can be released because the active site of the enzyme is actually targets the fatty acid which is attached at position number 2 of phospholipid. On the other side, we have the activation of phospholipase C, which can, for example, act on phosphatidylinositol diphosphate, thus removing, as you know, phospholipase C removes the polar part of the molecule. So it removes the alcohol component and it removes the phosphate group and leaves the acylglycerol as the product. So 1,2-deacylglycerol. Later on, 1,2-deacylglycerol can be subjected to the action of numerous lipases. So as you can see here, by the action of certain deacylglycerol lipases, DAG can be converted directly to release arachidonic acid and to give monoacylglycerol. Monoacylglycerol can also be subjected to the action of monoacylglycerol lipase to produce arachidonic acid, which you can see from the figure. So once arachidonic acid is released from membrane phospholipids into the cytosol, it can be converted to different eicosanoid molecules by a variety of different enzymes which are tissue specific. So different eicosanoids will be produced in different tissues and different cells. I'm going to give you just an example to illustrate this statement. For example, in vascular endothelium, PGE2 and PGI2 will be predominantly synthesized, so prostaglandins E2 and I2. But, for example, in thrombocytes, in platelets, primarily eicosanoid to be synthesized is going to be thromboxane A2 or AGTE, or hydroxyeicosanoic acid. So the molecule which is gonna be synthesized in a specific type of cells or specific tissues depends on the variety of different enzymes which are specific for that type of cell or that type of tissues. And this is summarized here on this slide. So this figure again illustratively represents the three major pathways which occurs in the biosynthesis of prostaglandins, leucotrienes and thromboxanes. So following the cyclooxygenase pathway, the first prostaglandin to be synthesized is going to be PGG2, and it can be further converted to either prostaglandins or thromboxanes. Arachidonic acid can be subjected to lipoxygenase pathway to produce hydroperoxyeicosanoic acid, which can be further converted to either leukotrienes or hydroxyeicosanoic acid or lipoxynes. And following the Cytochrome P450 pathways, arachidonic acid can be converted to produce epoxides and derivatives of HETE and HETE. So moving on to cyclooxygenase pathway, I would like first to discuss with you the structure of prostaglandins and to see which are the common parts of the structure of different types of prostaglandins, and then to see how we can give names to specific types of prostaglandins. So let's move on to cyclooxygenase pathway and begin with the structure of prostaglandins. So those are nothing else but fatty acids containing 20 carbon atoms, but this time with an internal 5 carbon or 5-membered ring in the structure, as you can see from this top figure. In addition to this very specific 5-membered ring, biologically active prostaglandins also have some different groups attached to the hydrocarbon chain. As you can notice from this figure, prostaglandins, all of them, have OH group at C15, positioned in alpha position, meaning below the ring. Then all prostaglandins have one double bond between C13 and C14. And as you can see, at C9 and C11, they can have different substituting groups attached to the 5-membered ring. Those attached groups are predominantly, as you can see from the bottom figure, are carbonyl group or hydroxyl group or double bond or hydroperoxy, endoperoxy group or some other. Depending on the types of those substituents in the ring, we have different types of prostaglandins marked with alphabet letters A, D, E, F, and so on. So for example, prostaglandins of the A group, they continue double bond between C10 and C11, and they have carbonyl group at C9. So whenever we say PGA, we know which substituents are found in the ring of those. Or PGD gives us the information that we have OH group at C9 and carbonyl group at C11. PGE class of prostaglandins, which are pretty active, and we're gonna mention them a lot today, they have carbonyl group oppositely from PGD, carbonyl at C9, hydroxyl at C11. PGF family of prostaglandins, they have both OH groups at C9 and C11, and so on and so on. So when we are giving the nomenclature and names for prostaglandins, first we start with PG, which is an abbreviation of prostaglandin, and then we add a capital letter from the alphabet stating which substituents are found in 5-membered ring. Then we give one more subscript, which is an Arabic numeral subscript, for example PGE1 or PGE2, which actually represents the belonging of that specific prostaglandin to one of 3 series of prostaglandins, and I'm going to explain later on in the next slide what does that mean, series 1, series 2, or series 3. Moreover, specifically for PGF family, we can have one more subscript available, which is a Greek letter subscript. For example, PGF 2 alpha is the most abundant. So 2 alpha means the position and the orientation of one additional OH group, which I'm going to explain later on. And here in the figure, you see these marks R7, R8, and so on. That means it indicates the number of carbon atoms which are left in that acyclic part of the molecule, non-ring part of the molecule. So we have 7 carbon atoms up and 8 carbon atoms down, and so on and so on. So what else can we say? Let me get you back to those series 1, 2, and 3 and explain why do we have those subscripts 1, 2, or 3 indicating the belonging to the series. So this very subscript that follows determines actually, as you can see here from the figure, the number of double bonds in the non-ring moiety of the molecule. So PGE, series 1 actually has 1 double bond, series 2, 2 double bonds, and series 3, 3 double bonds at specific positions. And of course, notice that these marks do not indicate the number of double bonds in internal 5-membered ring. So series number 1 has one double bond, as you can see here from the slide, between C13 and C14. And those prostaglandins of the series 1 are derived and synthesized from Eicosaptrienoic acid. Series 2 has two double bonds, as you can see from the slide, between C13 at C13 and at C5. So you will remember it easily because you always have C13 and then you have additional double bond in C5. And following the third series, it has three double bonds at position C13, C5 as previous molecules, and additional one at C17. Let me just show you here with the mouse at C17, as you can see from the bottom of the figure. Series number 2 is synthesized from arachidonic acid, and those molecules definitely predominates in the structure of prostaglandins and in our body. Series 1 and Series 3, which actually originates from eicosapentaenoic acid, those are produced to a much lesser extent because those polyunsaturated fatty acids are not that common in human diet. So actually today at this very lecture we're not going to focus on Series 1 and Series 3, so we're going to focus on those molecules belonging to the Series 2, which definitely predominate in our body. So let's summarize some other things. If you can see from the figure, C13, C14 double bond, its orientation is trans, so the configuration is trans. Other additional double bonds at C5 and C17 are of cis configuration. So I have already mentioned the Greek letter subscript, which is found only in PGF series, actually refers to the orientation and the position of the hydroxyl group at C9, and usually that position is alpha, which means below the plane of the ring. Speaking of the structure of thromboxanes, those are molecules which are derived from arachidonic acid by cyclooxygenase pathway, and they are pretty similar to prostaglandins in the structure, but there are some significant differences as well. So instead of 5-membered ring, as you can see here from the figure, thromboxanes have 6-membered ring with oxygen in the ring, and one more oxygen, let's say, bridge between C9 and C11. And again, we have C13 double bond and C5 double bond, which is the same as for PGs. Thromboxanes are named for their action in producing thrombi or blood clots, and the most common thromboxanes which we are going to deal with today is thromboxane A2, TXA2. So how those prostaglandins and thromboxanes are synthesized, that is the question which we are going to answer right now. So as I have said previously, we're only going to discuss about the biosynthesis of those PGs which are derived from arachidonic acid. So those are molecules which belong to the series number 2. So we're going to discuss about PGE2, PGI2, and TXA2. So let's start with the beginning. So I think that figure always gives us, and it's worth 1,000 words. So this is a nice figure taken from Mark's Biochemistry, which nicely explains what is going on when PGs or TXs are synthesized. So let's follow the metabolism of arachidonic acid. So the initial step which occurs with arachidonic acid is the action of cyclooxygenase itself. So this first conversion is catalyzed by cyclooxygenase enzyme, which means that we're going to incorporate some additional oxygen atoms in the structure of arachidonic acid. So if we follow the pattern here, we see that 2 oxygen atoms are incorporated as a peroxide group, endoperoxide group between C9 and C11, as you can see here, and 2 more oxygen atoms are incorporated at C15, forming hydroperoxy group. With these modifications and incorporations of oxygen atoms, the first prostaglandin PGG2 is formed, which is one pretty unstable endoperoxide, which is readily and quickly converted to PGH2, also as endoperoxide group, but this time this unstable hydroperoxy group is readily reduced to hydroxy group by the action of an enzyme, peroxidase, which you know from the previous chapter requires the action of reduced glutathione, which is going to be an acceptor of reducing, the donor of reducing equivalents, thus converting to oxidized form of glutathione. So now from the PGH2, which is the second prostaglandin to be formed, so from this molecule on, we can have different pathways and different prostaglandins synthesized. So let's follow the pattern starting from the left part of this figure. So from PGH2, PGE2 and PGD2 can be synthesized by the action of specific enzymes, PGE synthase or synthetase, or PGD synthase or synthetase. Further on, following the figure, PGE2 can be reduced by PGE9 ketoreductase to form the F family of PGs, so PGF2 alpha. This molecule, PGF2 alpha, can also be formed directly from PGH2 by the action of one specific endoperoxide reductase. So you can get PGF2 alpha, which is not represented here in the figure, directly from PGH2 by endoperoxide reductase. Following the next pathway, PGH2 can be converted to thromboxane A2 by the enzyme thromboxane A synthetase. The high concentration of this enzyme is found in thrombocytes or platelets, and TXA2 is responsible for platelet aggregation and causes vasoconstriction. On the other side, PGH2 can be converted to prostacyclin PGI2 by PGI synthetase, and this enzyme and prostacyclin PGI2 is produced by vascular endothelial cells, and it exhibits an opposite antagonistic action comparing to thromboxane A2. So it inhibits platelet aggregation and causes vasodilation. So this is a nice figure representing the metabolic pathways during the biosynthesis of different types of PGs. What are the functions of PGs and TXs? So here are some of the functions listed. I know that at this very part of your course you're not familiar with all of these processes as molecules, so please just remember those functions, and you're gonna hear more detailed story about some of those specific molecules later on. So the function of PGs is, for example, for PGI2, E2, and D2, increase of vasodilation and increase of the production of cyclic AMP. Inhibition of platelet aggregation, inhibition of leukocyte aggregation, inhibition of release of interleukins 1 and T cell proliferation, and lymphocyte migration. PGF2 alpha increases vasoconstriction, bronchoconstriction, and smooth muscle contractions. Some of the functions of TXA2 is an increase of vasoconstriction, so oppositely to PGI2, E2, and D2, platelet aggregation, lymphocyte proliferation, and bronchoconstriction. So it acts as an antagonist to those. But I would like to tell you something which you might maybe find interesting regarding cyclooxygenase enzyme, which is at the same time the major regulatory enzyme of the pathway. Actually, in the 1990s, it was found that cyclooxygenase is found in two isoenzyme forms, like cyclooxygenase 1 and cyclooxygenase 2, COX1 and COX2. COX1 enzyme is regarded as a constitutive form of an enzyme, and it's really widely expressed in almost all tissues. So its action is actually focused on the production of PGs and TXs as a normal physiological function. So it's not increased when it comes to inflammation or tissue injury. Regardless, this one, the other one oppositely to it, COX2, is an inducible form of the enzyme, and it's regulated by, of course, by a variety of cytokines and growth factors. So it's involved in it's affected by the cytokines, and it much alters the immune response of the system and the body, and it's usually not that active in healthy tissues and healthy body, but it's activated, and its activity is significantly increased when you have an inflammation in the body. So why is that so important, and why I'm doing this is because of the importance of PGs in mediating the inflammation response of our body. It was suggested that synthesizing certain medicines or drugs that can block PG production should actually provide us certain relief from pain, thus exhibiting analgesic effects. So definitely those both COX isoenzymes are inhibited by all non-steroidal anti-inflammatory drugs, or NSAIDs, and I know that some of those are so familiar to you. Acetylsalicylic acid or aspirin, acetaminophen, ibuprofen, those are all molecules which belong to NSAIDs, and they are capable to inhibit cyclooxygenase activity. So here we have a figure representing the mechanism of action of acetylsalicylic acid or aspirin, and it exhibits its inhibitory effect by transferring the acetyl group, as you can see here from the figure, from aspirin to active site of cyclooxygenase to OH group of serine from the active site. So the active site thus becomes acetylated, and it's irreversibly inactivated. Other NSAIDs like acetaminophen or paracetamol or ibuprofen, with the structures shown in the figure, actually act as reversible inhibitors of cyclooxygenase activity. Those NSAIDs are capable to inhibit and block the action of both isoenzymes, so both COX1 and COX2. Aspirin has found its application in the prevention of acute myocardial infarction by blocking the production of TXA2 from its major precursor, arachidonic acid. So by causing this mild hemostatic defect, low doses of aspirin has been shown to be effective for this purpose. I would like to continue the story on COX-1 and COX-2. When elucidating the actions and functions of those COX-1 and COX-2 isoenzymes of cyclooxygenase, people figured out that it could be useful to synthesize and to develop a selective COX-2 inhibitors. And there was a suggestion and a proposal that they could act as pretty potent anti-inflammatory agents by inhibiting selectively COX-2 activity, but this time, very important, without adverse effect like gastrointestinal disturbances like stomach ulcers or antiplatelet side effects, which are common adverse effects of NSAIDs. So, usually those adverse effects of NSAIDs are a consequence of the inhibition of COX-1, but selectively inhibiting COX-2, people found that they can achieve the pharmacological action, thus avoiding the presentation of those adverse effects. But later on, when those selective COX-2 inhibitors were used very intensively, it was found that the prolonged usage of specific COX-2 inhibitors may have a negative effect on cardiovascular function. And people try to explain what is actually going on in such cases. So it has been found that a long-term use of COX-2 inhibitors actually alters the equilibrium between PGs, for example, PGI2 prostacyclin and TXA2, and the equilibrium actually shifts towards the thrombic pathway, thus increasing the production of thrombi and aggregation of platelets. So actually after that finding, most of the COX-2 inhibitors are removed from the market, but there are still a certain number of selective COX-2 inhibitors which remain on the market, but they have to be and there must be used with caution, excuse me, and they are actually contraindicated with patients with ischemic heart disease or stroke. So this is something that you just have to have in mind. So COX-2 inhibitors were very popular at the very end of last century and at the beginning of 21st century, but then it was found that they exhibit numerous adverse effects. Some of them are withdrawn from the market, and the remaining COX-2 inhibitors have to be used with caution. And of course, they can be used, and they are still used in the therapy of different diseases. So, moving on to inactivation of those. So, I have mentioned at the very beginning that those PGs and TXs and Acorsinoids in general are readily inactivated. So, their half-lives range from a couple of seconds to a couple of minutes, and then they are subjected to certain chemical modifications which yields up their inactivation. So, PGs are inactivated by a couple of transformations. So first you have the oxidation of 15-COH group, and then you have the reduction of the double bond at C13, and then you have a couple of beta and omega oxidation of the non-ring portions of the molecule, and it ends up in the formation of decarboxylic acids, which can be excreted by kidneys and in urine. TXs or TXA2 is rapidly metabolized to TXB2 by the cleavage of the oxygen bridge between C9 and C11 to form two OH groups. And this molecule, modified molecule TXB2 is biologically absolutely inactive. And in order to, to close the story on cyclooxygenase pathway, I would like to give you one more, let's say, fun fact about those. I'm going to remind you of something that you can see and you can read from numerous portals and newspapers and magazines and so on, and everybody is talking about that. And that is the diet that include cold water fish, like salmon, like brook trout, like mackerel, like herring. So those fish, they actually have a pretty high content of polyunsaturated fatty acids, especially EPA, or eicosapentaenoic acid, and docosahexaenoic acid, DHA. Probably you have heard about those EPA and DHA fatty acids. So people who are practicing this kind of diet, they're going to have a pretty high content of polyunsaturated fatty acids in their membrane phospholipids. So it is suggested that such a diet can be potentially pretty effective in preventing heart diseases and other cardiovascular diseases, predominantly because of the fact that those are molecules belonging to that series 3 and series 1, if you remember from a couple of minutes ago. So actually, for example, instead of TXA2, which promotes the aggregation of platelets and formation of thrombi, TXA3 is predominantly synthesized, which is less effective than TXA2, thus probably active prophylactic to prevent the formation of thrombi and atherosclerosis and potentially myocardial infarctions, cerebrovascular infarctions, and other cardiovascular and cerebrovascular problems and diseases. And that was the story on cyclooxygenase pathway and some of those interesting moments regarding this pathway and PGs and TXs. But we have to move on further to lipoxygenase pathway. So lipoxygenase pathway also begins with arachidonic acid as the major precursor. And it's a substrate for enzymes which are belonging to the group of lipoxygenases. So there are more enzymes which are tissue specific, and the role and catalytic function of those lipoxygenase enzyme is the incorporation of an oxygen molecule into certain carbon atoms. So I would like to present you the figure which actually represents the transformation of arachidonic acid in lipoxygenase pathway. So I have mentioned that there are several types of lipoxygenases which are tissue specific, and we have 5 lipoxygenase, 12 lipoxygenase, and 15 lipoxygenase, and those names are given according to the position and the carbon atom to which hydroperoxy group is going to be attached. So if you can see from the figure, hydroperoxy group will be attached to the carbon atom number 5 by 5 lipoxygenase, to C12 by 12 lipoxygenase, at C15 by 15 lipoxygenase. So this is the step number 1, attachment of hydroperoxy group to these specific positions. Those enzymes 5, 12 or 15 lipoxygenase are found and are to be active in different tissues. For example, in polymorphonuclear leukocytes, 5-lipoxygenase isoenzyme predominates. In platelets, 12-lipoxygenase predominates. And in eosinophils, 15-lipoxygenase predominates. So those are cell-specific or tissue-specific. As a result of incorporation of hydroperoxy group at different positions, we have the formation of different 5, 12 or 15 hydroperoxy eicosanoic acid. So the name indicates hydroperoxy group, HOO group. Eicosa means 20 carbon atoms. Tetraene means 4 double bonds. And of course acid as an indication of the presence of carboxyl group in the molecule. So we have 5 HPT, 12 HPT, and 15 HPT. They can be further converted to more stable HET, 5, 12, and 15 HET or hydroxy eicosanoic acids, as represented here in the figure. What else can we say? From these molecules, HPTs, further leukotrienes can be synthesized, and I would like you to show and represent the figure. So let's start from arachidonic acid and its conversion to 5-HPT by 5-lipoxygenase. So the biosynthesis of leukotrienes begins with the formation of 5-HPT. So this HPT can be further converted to either HET that we have already seen or leukotrienes. In this case, first molecule to be synthesized is going to be leukotriene A2, as you can see here from the slide. So how can we form the names of leukotrienes? So they are synthesized in leukocytes, so that is why we call them leukotrienes. And triene, it indicates that each molecule has 3 conjugated double bonds of the trans configuration at C7, C9, and C11. So that is how our name indicates. So leukotriene means that it's synthesized in leukocytes, and its 3-ene indicates the presence of 3 conjugated double bonds. So in leukocytes and mast cells, we have the transformation of 5-HPT to epoxide form here between C5 and C6, and this is the molecule of leukotriene A4. This subscript 4 actually refers to the total number of 4 double bonds of leukotrienes. So as you can see here from the slide, 3 of those are, as we have said, conjugated, and that is why we have the name triene, and the last one at C14 is of cis configuration. What else can we say? Other leukotrienes can be further synthesized from LTA4 by 2 pathways. So the first pathway here on the right, so LTA4 can be converted to LTB4, which is kind of it's 5 and 12, I think, 5,12 dehydroxy derivative of LTA4. Or we can have another pathway which is slightly more complicated. So LTA4 can be subjected to the addition of the complete glutathione molecule, and the attachment of glutathione molecule at C6 is actually catalyzed by the enzyme glutathione S transferase. What else can we have? So now glutathione is attached to C6 of the molecule, and then we have to remove the glutamate moiety of glutathione by gamma-glutamyl transpeptidase, and then LTD4 is formed. And then LTD4 is subjected to the removal of glycine moiety by the action of an enzyme depeptidase to form leukotriene E4. So LTA4 can be converted to LTC4 by incorporation of glutathione. The removal of gamma-glutamyl moiety from the molecule yields the formation of LTD4, and removal of glycine moiety of the molecule yields the formation of LTE4 molecule. And different leukotrienes are synthesized in our organism. Speaking of the structure of the functions of leukotrienes, LTB4 increases vascular permeability, T cell proliferation, leukocyte aggregation, and expression of release of interferon gamma and interleukins 1 and 2. LTC4 and LTD4 increases bronchoconstriction, vascular permeability, and release of interferons gamma. I would like to pay attention to one more type of leukotriene molecules, and those are lipoxynes, and they are formed, as you can see here from the figure, by the action of 15 lipoxygenase followed by the activity of 5 lipoxygenase, of course on arachidonic acid as a precursor, and then a series of reductions occurs. So the hydroperoxy group actually forms 3 hydroxyl derivatives, which are known as lipoxynes, and this is lipoxyne A4, LXA4. Those LXA4 molecules induce chemotaxis and stimulate superoxide and ion radical production in leukocytes. But I'm not gonna give you more information about this class of molecules. It will really overcome the majority of the aim of today's lecture. And that would be the lipoxygenase pathway. Moving on to cytochrome P450 pathway, which actually means the activity of the group of monooxygenases in the microsomal system, which actually yields the production of different molecules like epoxides or hydroxy derivatives of AGTs or diol forms of AGTs. With the biological functions which is believed to have in ocular, vascular, endocrine and renal systems. And there are some suggestions that some of these activities and functions are based on the inhibitory action to sodium-potassium ATPase. But as I have said at the very beginning, the exact biological functions of those are still to be elucidated. And now I would like to tell you a couple of words about other Acorsinoid molecules apart from those 3 major groups. And I have mentioned at the very beginning the molecule of isoprostane, which is obtained and produced from arachidonic acid in the process of lipid peroxidation, which is an oxidative change of cell membranes and phospholipids of cell membranes initiated and caused by free radicals. And I'm sure that you have heard about these phenomena of free radicals and lipid peroxidation during your previous lectures. So those are changes and transformations which are not catalyzed by enzymes, but still occurs catalyzed by the action of free radicals and the extreme reactivity of those particles. So actually, during the oxidative changes of phospholipids, arachidonic acid is also changed, but it's still a component of phospholipids and exists in its changed and oxidized form. So phospholipase A2 normally removes the fatty acid from the position number 2, so we have the release of altered and changed arachidonic acid from the phospholipid, as you can see here from the figure. So the lysophospholipid remains in the cell membrane, and altered arachidonic acid called isoprostane is released in the cytosol and further on into circulation and in urine. So the level of those isoprostanes, as I have already mentioned, in blood and urine is used to predict the level of oxidative stress in the body of that specific patient. It is believed that those altered molecules of arachidonic acid also exhibit certain biological functions, but still those functions are investigated only on cultured cells and not in vivo. So it's yet to be characterized, and it's yet to be that different functions are going to be attributed to this group of molecules. And of course we have one more interesting group of Acorsinoids, which are endocannabinoids. So those are endogenously synthesized molecules which can be ligands, or they can attach and bind to cannabinoid receptors CB1 and CB3, and the primary effect of those is at the central nervous system. Among those endocannabinoids, the first one which was isolated and identified was anandamide, which is synthesized in neurons from phosphatidylethanol amine. And why is that so? Because I have mentioned before, it is actually synthesized by transferring arachidonic acid from the position 2 on phospholipid to ethanol amine moiety of the molecule. And I'm going to give you the figure here. As you can see here, so by transacetylase, so arachidonic acid is transferred from the position number 2 to ethanol amine moiety of the phospholipid molecule, and then using the phospholipase D, you have already heard about this enzyme, we learn that phospholipase D is capable to cleave ethanol amine from the phospholipid, leaving the lysophosphatidic acid and anandamide molecule. What else can we say? Once this molecule anandamide is released, it can act as a messenger, and it can bind to the receptors of presynaptic membrane, and it can, as a consequence, it can alter the ion fluxes, and it can affect the neurotransmitter release from the presynaptic neuron, and this can be increased and analgesic effect obtained. As you can see further on, anandamide can be degraded by fatty acid amide hydrolase, and actually this enzyme, as you can see from the figure, splits or cleaves anandamide to arachidonic acid and ethanol amine. This enzyme is interesting, and that is why I'm focusing on it. This fatty acid amide hydrolase here at the very bottom of the figure, actually it is nowadays it is a kind of a target of scientific research for new medicines and new drugs, because if we succeed to inhibit the action of this enzyme, we can keep and prolong the existence of anandamide in our body, in a human body, thus inducing the prolonged analgesic effect. So it's kind of a fun fact regarding those endocannabinoids. Moving on further, I would like to tell you a couple of things about the mechanism of action of those molecules. So definitely they exhibit a wide variety of physiological effects, and all of them are actually initiated through binding of specific Acorsinoid to a specific receptor on a cell membrane of a target cell. And then the mechanism of action is pretty similar to those mechanisms of regulation that we have already had as the regulation mechanisms for most of the metabolic pathways that we have discussed before. So when Acorsinoids binds to a receptor, it can do different things. So it can activate adenylate cyclase system to increase the production of CAMP, and CAMP affects the activation of protein kinase A to A system, and then it increases the phosphorylation of enzymes, which we have mentioned to be regulatory of those pathways. And those mechanism of activation is found in the processes and pathways synthesizing prostaglandins. And you have another pathway, so like we have already mentioned, we can increase that binding to the receptor, can increase the level of calcium in cytosol of the target cell, and then calcium can also affect the protein kinase A to initiate the phosphorylation and activation of those. And this mechanism of regulation is actually found in PGF2 alpha, thromboxane A2, and peroxide and leukotrienes. Some of those actions, let me get back to your knowledge on the regulation of metabolic pathways. So it can be achieved through the activation of adenylate cyclase system as a response to the certain stimuli and binding of certain molecule to a specific receptor. So one more thing can occur here, and you know that it can yield to the activation of G proteins, which are found within the plasma membrane of the target cell. And you know that there are two moieties of G proteins. So if the Acorsinoid actually binds to the stimulatory subunit, the effect is going to be increased or amplified. But if it binds to inhibitory subunit, the cellular response will be reduced or inhibited. So that is kind of of the mechanism of action through G proteins and adenylate cyclase system. But as I have mentioned at the very beginning, some of those molecules can act as we said, local hormones, which means that they can exhibit both paracrine or autocrine action. So let's define those. I'm going to give you an example of paracrine action. So the concentration, for example, as I have written here, of vascular smooth muscle cells caused by TXA2 released from circulating platelets, causing vasoconstriction. So TXA2 is synthesized in thrombocytes, but its action is on vascular smooth muscle cells. Or an example of autocrine action of Acorsinoids, TXA2, which is synthesized in thrombocytes, affects the platelet aggregation, so at the site of its production. And what can I say as a conclusion to those is that Acorsinoids in general definitely affects and influence the cellular function of almost every tissue of the body. And of course, like everything, certain organ systems are affected to a greater degree than others. And to conclude today's lecture, I would like to give you a couple of examples, because without examples and without the possible application of those theoretical facts and data, I think that those facts are going to be kind of useless. So we have to find a purpose for giving you that many information like today. So I'm going to give you a couple of examples. Some of those we have already mentioned. So example number one, we mentioned the prevention of the platelet aggregation by usage of the low doses of aspirin. So that is the first example of the exact application of this knowledge in medical practice. So in the presence of aspirin, as we have said before, cyclooxygenase is irreversibly inactivated by acetylation, transfer of acetyl group from aspirin to the active site of cyclooxygenase. So, the new molecules, the novel molecules of COX are not going to be synthesized. Why is that so? Because thrombocytes, they don't have nucleus. So they cannot provide additional messenger RNAs for the synthesis of cyclooxygenase enzymes. So the effect lasts as long as the lifespan of thrombocytes, which is like 7 to 10 days. So when aspirin is taken daily at low dosages, those dosages ranges between, I think, 75 to 150 milligrams a day in most cases. So the new thrombocytes, the new platelets are also affected by blocking the COX enzyme, and actually the effect is transferred to new platelets as well. The higher dosages of aspirins are definitely not recommended, and they do not improve efficacy, but do increase the side effects such as gastrointestinal bleeding or being bruised easily, and so on and so on. But low dosages are really common and pretty effective. And I know a lot of people, for example, that I know that are taking regularly low dosages of aspirin in the prevention of the platelet aggregation, formation of thrombi, plaques, atherosclerosis, and consequently cardiovascular diseases. The second example that we have already mentioned at the very beginning is the application of glucocorticoids as for purpose to reduce the inflammation in the tissue, both acute and chronic inflammation. So corticosteroids in general can reduce inflammation by inhibiting, if you remember, by inhibiting the action of phospholipase A2, which prevents the release of arachidonic acid and prevents the increased production of PGs and T-axis. So anyway, glucocorticoids are also believed to suppress the activity of COX-2 isoenzyme and thus exhibiting additional anti-inflammatory activity. But regardless the fact that glucocorticoids are really valuable in pharmacotherapy for treatment of both acute and chronic inflammation, they tend to exhibit certain severe side effects, and for example, they can yield to the sudden appearance of glucose intolerance if used in pharmacological doses for extended period. But what is the value of the application of glucocorticoids is if they are applied as inhaled glucocorticoids. So they have a much fewer side effects or systemic side effects than those which are absorbed through gastrointestinal tract or given directly into circulation through injections and so on. Why? Because the actually absorption of those inhaled glucocorticoids through bronchial mucosa is pretty limited. So their action is limited and focused on the site of application. So the patients who are suffering from asthma, they can safely use glucocorticoids which are inhaled. So you're gonna, they're delivering glucocorticoid direct to the site of inflammation, thus not affecting the other systems of tissues and organs, and of course thus increasing the effectiveness of those glucocorticoids itself. The third example of the possible application of this knowledge is that the analogs of PGE2 and PGE1 are actually used as drugs which are used to suppress gastric ulceration, in part by inhibiting the secretion of hydrochloric acid from the epithelial cells of the stomach. So PGE1 and PGE2 without any kind of gastric ulceration. The fourth example, some analogs of PGE1, while we are still with PGE series, PGE1 can be used for the treatment of sexual impotence. So men with certain forms of sexual impotence can self-inject the analog of PGE1 directly into the corpus cavernosum of the penis, thus initiating the immediate but of course temporarily penile tumescence, and the erection actually lasts for 1 to 3 hours. Also, PGE2, while we are still with PGE, and PGF2 alpha family, which actually causes the construction of smooth muscles, as if you remember from our previous stories, that stimulatory action of those two on uterine muscle contraction actually ended up in the usage of those to induce labor and to control postpartum bleeding. And I'm going to give you a few more examples. PGE, excuse me. So anyway, we had a little bit of problems because I ran out of the battery. So that was all that I wanted to tell you about prostaglandins, thromboxanes, and leucotrienes. I hope that you could follow me and that you could understand the story, and I also hope that the story was kind of useful and interesting. So until some other topics and some other stories, stay safe, stay healthy, and bye bye.